briefly, since most of you know Brian quite well. Brian got his PhD from Arizona. Brian got his PhD from Arizona, University of Arizona. I think I might have even overlapped with him for a little bit. And then he did a postdoc with Kamai at University of Washington and UC Davis. And it was interesting. I'll just tell a really quick story. We had to do a site visit because they had a big grant. And Luca was one of the people. And so we grilled them with questions. And these site visits are pretty grilly, you know, and Jimmy Walbert was there, so it was very really. And you know, the response, the most common response is, gee, I don't know, I'll have to ask Brian. So Brian was clearly kind of helping to run the show there. And I think that's just an indication of his enthusiasm and knowledge. And I think that will be on display today. And one last anecdote. So I have a whiteboard, which I never use, but every time Brian stops by, we'll get to talking and he'll write on the whiteboard. And so I finally just wrote Brian's board on the whiteboard because he's always got something to say, as he will today. So with that, welcome, Brian.
So I'll tell a different story about that exact site visit, because there's something that actually Damon said to me at that site visit that really influenced the way I have tried to be a scientist here at Purdue. I had, at the time of that site visit, I had received the job here, but I hadn't yet started. And so it was like my last act was to leave the polyploidy project in good health and jet off to Indiana. And Damon had an aside with me, and he said, Brian, can you publish papers in Genetics, the journal? And I said, yeah, sure. And he said, okay, I don't want to hear about you freaking out. Because Vicki Chandler made it into the National Academy on the basis of her work in genetics. So clearly that's enough. And it's something that I have replayed in my head multiple times over the years. It was a very nice thing to say, and it was a very true thing to say about Vicki Chandler. And it turns out I can publish papers in genetics occasionally, although the House journal is now G3. Okay, and with that, let's talk about some problems that are as old as life itself. We're going to take a look at a bunch of different systems approaches to really looking at genetic interactions and genetic mechanism. Almost all the examples I'm going to give today are in maize because it is now my favorite study organism. But we're going to talk about things from the atomic level all the way to the genome level and maybe a little bit of field scale. So since you never know when you're going to get cut off due to time, I just want to say the work that I'm going to present today, this is not a complete list of our collaborators. We're a very collaborative lab, but everybody that's underlined here contributed some analysis or data to the slides that I generated today. And then the work was done collaboratively with this group of people. And I just want to call out Damon. There's a project that we'll get to that is involved him, and of course 2 very talented postdocs in the lab, Rajdeep Kangura and Aman Kaur, whose work I'm going to talk about extensively. One of the project was started by an undergraduate named Elizabeth Mahood, which many of you met when she interviewed here for a graduate position. She went on to Cornell and is now at MIT doing the AI of protein folding, as it turns out. Lauren Witt, another graduate student whose work I'm going to talk about quite a bit. I just want to call out, and then just because I'm gonna pass her by, not spend that long on it. Irina Makarevich is a PI at Hamline University. It's a primarily undergraduate institution, and she's been working on taking a bunch of stuff that we do in the lab and turning them into cures or classroom-based undergraduate research experiences. And I think that's really, really exciting, and that's a result of funding from the National Science Foundation. Okay. So I love carbon-based life forms. I hope you love carbon-based life forms too. But I think more people should love carbon-based life forms. And this is a graphical representation of global biomass distributed by taxa. It's worth saying, just because it's true, there's more carbon in Rubisco than there is in humans. And humans are the single largest single species biomass on the planet. So I really think there's no reason to study humans. They're boring. They don't do anything interesting. Plants are where it's at. If you care about carbon-based life forms, you'd work on this, not some rounding error on some blow up that was made to make us feel better about ourselves. All right. Yeah, there's 5-fold more carbon in Rubisco than in humans. Okay, so my name is Brian. It's been 0 days since I started a new project. I'm actually not kidding. Chris Oakley and I started something yesterday while we were at coffee. He's laughing because it's true. This is the downside of being interested in carbon-based life forms. I've never been faulted for lack of curiosity, but the good news is that there are lab-based treatments for my condition, mostly getting some work done, and with consistent application of new data, I have been able to lead a healthy and productive life, which I hope I will demonstrate for you today. I like to stare into the abyss and try and construct meaning from the nothingness with questions. I am full of questions all the time. I love questions. I also like phrasing them as declarative statements so that they can be hypotheses and they can be tested, right? But questions are really what get me going and I love. I would like to be better at, I wish to achieve that my lab will prepare and deposit results and methods in ways that allow others to engage in reuse. We do a lot of reuse of pre-existing data. We ask new questions with old observations. I love doing that. I love it when other people do that with our stuff. And so it's something that matters to me a lot. And I often think of the fictional character, Count Tyrone Rungan, as he is torturing the man in black. And he says, this is for posterity, so please be honest. And that's sometimes how it feels when you're trying to get your stuff out into a database. But it really does matter because everyone is your collaborator. If you're data sharing, then everything is a collaborative project. Everyone is your buddy. There's no such thing as being scooped. You're simply permitting data reuse and the onward march of humanity. One way of phrasing this is, before I started this job, Sam Hazen, who's now at the University of Massachusetts, and I were having a conversation. And I said, what makes you think we're going to be able to solve any of these problems? And his reply was, my friends are smarter than your friends. Which again was a very sweet thing to say, because I am his friend, and he is my friend. Anyway. Took you a minute. Anyway, all right. So genetics, to me, is the atom smasher of biology. We put in these very simple binary hypotheses. There is a gene linked to this SNP or not, and you get a result that says yes or no. And so it's a very structure function relationship. It's extremely tight and tidy. I love it. My particular view is I look at this with a very explicitly evolutionary perspective. I am interested in proximate causality. I want to know what's causing this thing to do that. And so that's molecular mechanism. I also am very mathy. I am a quantitative person. If you can put it into a number, it's better. As far as I'm concerned, always. Phenotypes are treated quantitatively. Alleles are treated quantitatively. How much it costs, how much of your time it takes. Could you do it more accurately? You know, so the question of can you do that cheaper, faster, better, or smarter gets asked a lot. And the downside is if you can do it cheaper, we can do more of it, right? So it's never a matter of like, let's do less. It's always let's do more. Let's keep going. So directly, physically, explicitly, this is a natural system. We are natural scientists. This is why I think molecular mechanism is so exciting, is it keeps us anchored in the real world. It has to work in the real world or we're out. Okay, so here's an old problem. Here's an awesome old problem. That's Alfred Russel Wallace, one of the two people credited with the discovery of the theory of evolution. And he made an observation in 1895 that there's nothing more abrupt than a change, often due to the diversity of soil. A sharp line dividing a pine or heather-clad moor from calcareous hills. So if you've ever gone for a walk outside, and especially at a time like now when the colors are really clear, and seen sections of green and sections of yellow, big changes in species diversity or in plant performance, like you can see here with these yellow flowering varieties or species in an otherwise green grassland, that's due to the erosion of these ultramafic rocks, creating serpentine soils that are extremely toxic, high levels of heavy metals. And you know, it's not just natural beauty, it's also what's not for dinner. This is a rice field that has levels of salinity that are high enough to reduce yield to zero in large sections of it. And so this ability to adapt to different soil systems and to different elements, right? So to the accumulation of different elements. These are, again, atoms that are present in the environment that plants need to use or exclude if they are bad for performance. And this is a very old problem. Keeping your insides in and your outsides out is what made life happen, right? So the compartmentalization of the living thing as distinct from its environment was a necessary component in the very, very fundamental evolution of life. So metabolism requires essential minerals from the environment, such as iron for energy transfer reactions. These things are regulated very tightly in a manner that's best described as homeostatic. So there's homeostasis for these things, which means it's a very active biological process going on. It also does this through metabolism. It's not just necessary for metabolism, but organisms produce metabolites that are involved in sequestering, modifying, or transporting these elements. It also is necessary to exclude toxic stuff like arsenic, or like in that serpentine soil example where there would be high levels of nickel. And these are also seen as homeostatic and will affect each other. Everything is linked. It's a living system. It's an iterative system. It's engaged in this constantly. So this is going to involve all sorts of things that are very, very old, like membranes, mechanosensitive ion channels. And so there's going to be deep time conservation of the mechanisms that control this. There will also be species-specific stuff. Don't get me wrong. There's all sorts of reasons to study esoteric, cool stuff. But fundamentally, the problem of keeping bad stuff out and good stuff in is necessary for all life and is accomplished by the same machines across all of life. So one visualization of this was done actually in reaction to Ivan Baxter getting annoyed at me picking candidate genes in our GWAS experiments. So Lauren Witt, who's a graduate student in his lab, created a curated list of genes that affect the plant ionome. So let's just get all the genes that we know that can affect elemental accumulation. Let's put them in a list. Then let's identify the orthologs of all of those genes in a variety of species that people tend to work on. So in yellow are all the genes for which we have an experiment demonstrating that it affects the accumulation of elements in a plant. Most of those are from either Arabidopsis or rice. Everything else is inferred by comparative genomics. Everything else is a blast search, basically. And so this has awesome power, right? Because if it's a very old problem, like keeping heavy metals out, then a heavy metal ATPase is very likely to be involved in it. And it's going to be performing the same function in Arabidopsis and yeast and E. coli. So it's also going to be doing the same job in Ceteria. Like, this is not complicated. This is not some, oh my God, evolution. No. This is very, very simple, right? However, when we do a genome-wide association experiment, the first thing we do is try and identify alleles that we can explain, which means we simply rediscover the same stuff over and over again, which frankly is a little boring, right? It's nice to know that there's an allele in Ceteria italica at this particular HMA that is involved in excluding cadmium, because people do eat Ceteria and it would be useful for them to eat less cadmium, but it's not especially exciting from a biological point of view. So allele discovery is definitely helped by this. And the other problem that we have in these genome-wide association experiments is the nervousness of the plant molecular biology community. So we do these genome-wide cutoffs, right? Everything above this line is almost certainly true. That's where the cutoff is. That doesn't mean that everything below the line is not true. In fact, a bunch of the stuff down here is true. It's just our certainty isn't high enough to allow reviewers to say, yes, you know what you're talking about. So there's a huge false negative problem here. And humans are horrible at thinking about false negative problems. I have made it my business. So we're going to talk about this and some ways to get around this that we've devised. So over the years, we've done this a bunch of different ways. Here's one. We used SNPs, elements, and transcripts, trying to identify genes that were co-expressed that were also linked to genome-wide association hits that were below this line, and used gene co-expression networks and subnetwork analysis to try and connect these things. And this guy, Bobby Schaefer, who is currently interviewing to be an astronaut, actually built this code and ran this project. And I'm not kidding, he really is. Awesomely, unexpectedly, and beautifully, we hit other stuff that the lab happens to work on, specifically GA signaling and the deledomain GRAS transcription factors, which we were already working on and had built large segregating populations to study. And so we were able to deliver seeds of this to Ivan, that's him picking it up in Indianapolis, and demonstrate that single locus mutants in D9 and its paralog D8 both affected the level of cadmium and other elements. So our gene co-expression, GWAS overlap worked. Take two, I'm just gonna say this crisply, take two orthogonal data types and overlap them, and you can play the trash to treasure game. And this works for anything. You just don't want to take non-orthogonal data types, because then you're getting high on your own supply. All right. So GWAS detects alleles, and alleles are relatively young, right? So if we have genes that are as old as life itself, our alleles are arising in the species at the tip here. They're population genetic phenomena. They happen inside each one of these endpoints on this tree. But the solution to the problem of keeping your insides in and your outsides out is before land plants for many genes. So maybe each one of these species is really a replicate. These are each orthogonal pieces of data. If I do a mapping experiment in rice and a mapping experiment in sorghum, and I find the same gene in both controlling cadmium levels, I have allowed evolution to carry out a replication experiment for me. Every tip from a population genetic point of view is a replay of the evolutionary tape. And this has occurred multiple times. There's lots of studies showing sodium accumulation is affected by HKT1, or molybdenum accumulation is affected by the molybdenum transporters, or copper levels are affected by the heavy metal ATPases. So this happens over and over again, and people just sort of casually said, yeah, no big surprise, it's the same stuff all the way across. Well, why don't we use that? Why don't we take that as an approach? So we, over the years, have been involved in measuring elemental accumulation in the seeds of a wide variety of flowering plants, including maize, sorghum, rice, Arabidopsis, and soybean. So what we did is we asked the question, are GWAS hits associated with genes that are orthologous across these 5 species? And if so, do they map to genes that we think are reasonable? Does it make sense? So we have that set of known ionomic genes, which we can use as our ground truth set. And we have some interesting possible questions that we can get at here. One thing just to call out, because a couple of these are from here at Purdue, so extra suberin 1. I don't know if anybody remembers Prashant or was on Prashant's committee, but Prashant cloned this gene, and it regulates lignin and suberin in the Casparian strip of Arabidopsis. It's never been demonstrated to be important anywhere else. Turns out it affects the level of calcium in 3 out of 5 of these species, natural alleles. So bang. Same with Brutus. It's an E3 ligase that regulates beta HLH proteins that are involved in iron homeostasis. It's involved in the levels of iron in 3 out of these 5 species. So as we dug in here, we've got 134 true positive natural alleles discovered simply by overlapping the known ionomic genes with our GWAS results in our 5 species, which is more than we would expect by random chance, by a lot. So it works, right? Turns out evolutionary tape idea, these are old problems. We should see new alleles in the same genes. That all checks out. But it's actually only 2% of all the GWAS peaks that we detect. So it means that even with all of the effort that's been expended by all of humanity on studying elemental homeostasis, we still have 98% of the world yet to discover. which I think is really exciting because I'm alive now and I'd like to do some science. The other problem is that our false detection rate is about 76%. So there's 134 true positives in there, but there's a huge amount of overlap. That's bad. So good, more than expected by chance, bad, high false detection rate. So, and of course, these are all known to affect the ionome already. So that may not be that interesting. So Lauren Witt built an approach that she calls filtering the results of multi-species analogous GWAS experiments, or FireMage, and she's a D&D fan. And what it does is it takes the intervals that are output by each of the GWAS experiments in each of the species, and it detects loci that are linked to those, and then carries out an orthology comparison. Are these true hits associated with orthologs? Then it takes the same number, creates 1,000 versions of that output, 1,000 sets of genomic loci of the same size, so a permuted fake set, and then looks at the genes in that interval and does the orthology comparisons. That gets us our expectation for how often this should happen by random chance, given these inputs, the inputs of our GWAS experiments. How often would we expect to see orthologs versus how often do we see it in reality? And by comparing that permuted data set to the true data set, we can figure out how well did biology enrich for orthologs. That gives us our false discovery rate. So we've done this a bunch of different ways, and we've had a lot of fun with it. And this is the upset plot that takes us to where we are now. So orthologous genes encode more GWAS loci than expected by chance by a lot. There is an interesting problem, which I'd love to talk with somebody who cares about it later, that synteny actually reduces the amount of signal here. So if you use species that are more unrelated, you get better performance. David says yes. You can come up with other ways of doing this that are easier, and they produce what I will refer to as a trivial null hypothesis. You will get an awesome performance of the system if you use a dumb way of doing this. So don't fall victim to trivial null hypotheses. Don't ask how often do this number of genes overlap. Ask how often do this number of intervals overlap. And this is why it's important to use genomic regions and not SNPs or random sets of genes or whatever. The unit of analysis here is genomes. The unit of analysis is not SNPs. The unit of analysis is not genes. This is about how genomes are related to each other. So, so this is awesome. We've had a great time. This is our FDR. So we get some improvement, but you can see that when we hit all 5 species, our false detection rate drops dramatically. And as we allow some species to drop out, this is our 5 out of 5 list, our 4 out of 5 here in peach, and our 3 species out of 5 here, the false detection rate begins to head back up towards where it was for the KIG at 76. So the more species you have, the more opportunities to look at that evolutionary tape, the more replication you have, the lower your false detection rate gets, which is absolutely expected. Damn.
What are the columns here? Is it
Number of overlaps. So there's a huge
different
orthologous genes with loci. So there's a huge number here at 3 out of 5 when you include Arabidopsis, Glycine, and sorghum, and that's a function of essentially the input pieces. But the false detection rate there is about 50%. So there's 1,700 overlaps, of which 800 are real. Is that John?
Is this an aggregation of all of the ionomic, all the data?
All 17 ions in all 5 species. It's a huge amount of data. That's a beautiful summary of a mountain of data, right? Done by Lauren. I think a really nice job. And for those of you who like Venn diagrams, I think this would be un-Venn diagrammable. So the upset plot wins again. All right. So we also observe that when we do, what happened? Oh no, I'm all right. I'll just keep coughing. Rather than spit that out on someone. So when we test Arabidopsis mutants that were identified by at genes that were identified by this, we see a high rate of conversion. So we get statistically significant differences in the elements that we expected. You can see here AT1G0300 with a change in the level of molybdenum as expected. Our 5 out of 5 list includes a number of things we already knew. MOT1 and MOT2 are both molybdenum transporters. That's what the MOT stands for. For metals, we got the heavy metal ATPases. For manganese, we got a transporter in the NRAMP family that we knew was a manganese transporter. And we got another one that really, I feel like a dummy, because if you go to bioRxiv and look at this paper as it is now, this is in there with a sort of like, I don't know what that is. That is SESA6, that is cellulose synthase A6. I previously published a paper on this with Chunhua, and I should have seen it coming. That's the gene that was the receptor of her ES20 drug. And even better, the trait is for manganese. And maybe some of you remembered this, but manganese is at the active site of SESA and required for its catalytic activity. Manganese is loaded into the vesicles where the rosettes are built and then transported to the plasma membrane. And I think it is reasonable to speculate that SESA may actually be involved in manganese homeostasis directly, either like ferritin as a large sink for manganese, or as a moonlighting as a sensor. If this is unloaded with manganese, maybe you need to turn your manganese transport system up because we need to make cellulose or we're gonna get eaten. So I'm really excited about this. And actually I talked to Chris Steiger recently and we swapped some seeds and we're gonna test whether this is in fact the case. Everything else here has checked out. So I'm expecting SESA6 to also have a manganese phenotype, which could be, I think, really exciting. Okay. So this had an advantage. Evolution is true. Alchemy is not real. All of our traits are exactly the same, no matter where we measure them. Iron is still iron, whether you find it in soybeans or Arabidopsis, but plant height might not be, right? So determinacy might be different than cell elongation. So how well is this going to work across other phenotypes? I'm very excited to learn, and we're going to do some experiments on reproductive maturity and plant height and see if we can go there. It does work for seed size. We've already done it because seed size is part of the measurement of the seed ionome. We've done seed size, and now it gives me the opportunity to talk about Irina Makarevich and her amazing work at Hamline University. So because we can do seed size, we can test Arabidopsis mutants in fire mage candidates using an undergraduate research experience that's done in a course where they grow Salk lines that are defective in one or another of the genes that we identified by fire mage as affecting seed size. They can then do some machine phenotyping of those seeds on flatbed scanners, which are cheap and very reproducible. They can get experience using plant CV to process those data and think about how the machine eye works, how the human interface with the machine works for getting very large numbers of data back for what is a relatively simple phenotype. The exercise is extendable. If students like it, they can carry it on and do more. But it gives them experience with growing plants, the concepts of phenomics, and the concepts of genetics. So it's a very nice exercise. So evolution is true, alchemy is false. Orthologous genes encode independently derived functional alleles detectable by multi-species GWAS. There you go. There's my little poem for you. That's the summary. And I want to move on to more things that are as old as life itself in the time that remains. Yes, I'm doing okay. So tetrapyroles, I love tetrapyroles. I think these are awesome. I have trouble spelling the word, but I think it's an awesome metabolite. So these are in everything that is alive. This is one of those few set of steps and metabolites that are in everything. And I want to point out that once again, plants are cooler because at this step here, protoporphyrin 9, you can either zig, add an iron, and you become heme, or you can zag, add a magnesium, and become chlorophyll. So plants, cooler, right? Just to be clear. All right. So this is exactly that spot. And these metabolites are great at moving chemical energy around. It's why they're at the core of photosynthesis and splitting water. It's why they're at the core of so many reactions. If you think about P450s or heme-dependent dioxygenases or any number of ways in which electron transport is achieved by heme, this is absolutely central to everything that we're doing right now. We're all dependent on this, and I'm so grateful that it's in us. All right, however, all technology is both good and bad, right? This means it's super good at moving chemical energy around, which means if you have the wrong kind of chemical energy, or you tune the machine incorrectly, it's extremely dangerous. And if you stop this pathway here, you will produce metabolites that are light-powered superoxide generators. They just kick off superoxide constantly. They will destroy your membranes. You will not be able to keep your insides in or your outsides out, and you will die. Bad. Same thing here. As you break down chlorophyll, it happens again. And you end up with these chlorophyll catabolism mutants that do the same thing. They can receive light, they then create superoxide, bad things happen to membranes, and down you go. For whatever reason, heme is easy. So, on the way out of heme, we don't have the same problems. So, as the great Oakland funk band Tower of Power pointed out, don't change horses in the middle of the stream. Red chlorophyll catabolite reductase is one of those steps on the way out of chlorophyll. If you disrupt this gene, it creates the accelerated cell death mutant in Arabidopsis or the drop dead 2 mutant in sorghum, which Jacob is working on. And you can see here, it makes these eye spot lesions. And if you stop at the step prior, that's actually in maize, that's called lethal leaf spot. That was the first lesion mutant cloned in any species, or accelerated cell death in 1 and drop dead 1 in sorghum. And here's the drop dead 1 mutant in sorghum, also cloned here. So bad things can happen when you try and bust that ring open and don't finish the job. Same thing goes on building it in the first place. And here is a mutant that this is maize. It's at 30 days after sowing. And you can see there's these little spots all over it and a slightly smaller number of spots here. So both of these are semi-dominant mutants in the LES28 gene. And for lesions 28, the 28th lesion forming mutant. And you can see we've got these regions of death that are caused by light-dependent superoxide production, membrane peroxidation, and death. This is just pure metabolic mayhem, just as simple as that. Nothing fancy here. And like I said, these are semi-dominant, and LES28-2, which is the weaker of the two, can be recovered as a homozygote, but LES28-1 cannot. You can imagine why this might be. If you can't make heme, you can't eat or make energy or make ATP or carry out most of the reactions in the cell, and you can't make it. So this one completely dead, but clearly has a semi-dominant phenotype. This one much weaker and can be recovered as a homozygote. That homozygote displays a pattern called diurnal banding. This is a pretty cool phenotype where we see the severity increase in a pattern, right? So brown, green, brown, green, brown, green, brown. And that pattern matches the path of that leaf out of the whorl and into the world. So the parts of the leaf that extend during the night show one phenotype. The parts of the leaf that extend during the day show another. And this is a diurnal pattern that can be disrupted by growing the plant in 24 hours continuous light in a growth chamber. The whole thing is just covered evenly in lesions. So this diurnal pattern requires that diurnal cycling of light. But interestingly, the lesions don't form immediately. So whatever this pattern is, it gets set up in the leaf, hangs out for like 4 or 5 days, and then kicks off. I have no explanation for this. No one else has any explanation for this. Nobody knows what's going on, and I'm not going to provide you with an answer. I think it's really cool that we have these absolutely obvious, beautiful patterns, have everything to do with the rotation of the Earth and the sun turning on and off, and we still have no idea how that works. But we can suppress all lesions by simply putting a piece of tinfoil over the leaf and preventing light from causing that superoxide. So it's light dependent, it's diurnal, it encodes uroporphyrin 3 synthase, which is one of those genes at the beginning of the tetrapyrrole pathway. If we make a double mutant, we can actually have a severe yellow phenotype when we combine these two, demonstrating that they were allelic and allowing us to say we got it. In the literature, it says less 22, and by extension, presumably less 28, is haploinsufficient. Haploinsufficiency is a super interesting, coming up on my time, super interesting phenomenon where only one copy of the gene is not sufficient to maintain wild type phenotype. However, no data was presented. They just said so. And the editors at Plant Cells said, cool. So we decided to test that because that seemed like an important thing to do. So the first thing to do, since less 28 and less 22 are on the same chromosome, the first thing to do was to demonstrate that if we make a double mutant between less 28 and less 22, we have severe lesions, slightly more severe than either mutant alone. So if we create a plant that has only one copy of less 28 and less 22, if this phenotype is haploinsufficient, the plant should look like that. And I knew before we did this experiment that it didn't, because monosomics, plants that only have one copy of one of the chromosomes, are well characterized in maize. They're available from the Stock Center. They're available from Jim Birchler at the University of Missouri, and they do not form lesions. Not only that, we can even see that there's changes in gene expression and that the number of transcripts goes down by more than half, even though these plants show no lesions whatsoever. So neither less 22 nor less 28 are haploinsufficient, nor less 2, which is also a lesion-forming mutant, also on chromosome 1. So simply, it's not true. They are not haploinsufficient. What they are is weirdo alleles. They have mutator inserted in them, and we have years of EMS mutagenesis that we've done. We've never recovered another allele of less 22 or less 28. All of the mutant alleles that are available contain a mutator transposon, indicating that it's the mutator element that is somehow responsible for the lesion-forming phenotype, and the monosomies have less than half the transcript and no lesion phenotype. So with that, because I think, oh no,
You've got plenty of time.
that's fat. I looked at the wrong piece. Awesome. I thought I took too long. I was like, holy shit, I'm never gonna get the.
The shorthand is
Go
the
ahead.
hour. And the long.
Yeah. Thanks. I've just spent too much time looking at my phone. That's how you know what's in front of the audience. All right. So less 28-1 is famous. It's an awesome mutant. It was Martinson and Barron in 1994 in the Journal of Genetics showed that this was a mu suppressible mutant. That means it shows epigenetic regulation of its phenotype that was dependent on the status of the mutator transposon. But this is where I'm going to lose everybody. Welcome to Maize Genetics. I promise we'll have a recap. All right, so what happens is you get this spontaneous silencing of the lesion-forming phenotype and these green leaves as the mutator transposon turns off. So mutator transposon turns off, weirdo allele at less 28 disappears, right? Again, fabulous result, but it means these alleles are responding to mutator activity somehow. The phenomenon of mu suppressibility is one that has been studied to differing degrees. So Damon went back, he made a bunch of crosses of less 28 to the active mutator line that contains an active mu-DR and his super special weirdo rearrangement called mu killer, and was able to show that that less 28-1 indeed not only is mu suppressible in 1994, but now that we have these click on, click off genetic tools, that this phenomena holds up in 2024. In addition, we made a similar set of crosses with less 28-2, which was the weak allele, which also has a mutator element in it, and it turns out to also be mu suppressible. If we if we silence mutator, we get a less severe lesion phenotype in less 28-2. And we see this in multiple successive generations. And that's nice. So basically, both of these alleles appear to be weirdos. Both of these alleles appear to be mu sensitive. So the transposon is inserted in them, and somehow that transposon has become part of their cis regulatory sequences, and that's important for the expression of the lesion phenotype. Which means they're reporters for mutator activity. So we now have an easy to score, like how many dots does it have? How light green is it? Easy to score phenotype for mutator activity. So we could basically, at 2 miles an hour, walk through a maze field and say, severe, weak, and phenotype anything we wanted to for mutator activity using less 28 as a reporter, right? It's a reporter for mutator activity. So we did a crazy experiment, and we've done a lot of these experiments. I think we're at 19 or 20 of them. I'm only going to talk about maybe 1 or 2 today. We have been crossing these dominant mutants of maize to an association panel. So that's, it's a few hundred crosses, no big deal. And then you grow families, the next generation, out. You start with a heterozygous tester. So your families are F1 hybrids that are wild type and F1 hybrids that are mutant, roughly 1-to-1. So each plot is then a case and control set that are isogenic except for the one mutant that you're interested in, or I'm interested in, because I didn't cross your favorite mutant. But if you gave me your favorite mutant, I might. And we can then use the genetics on the other side, the association panel, to carry out a genome-wide association for epistatic interaction of the genome-wide diversity of maize, only the most interesting species available, with whatever mutant we're interested in. So we're anchoring our understanding of natural variation by GWAS in the frame of what modifies less 28. So I've said mu, but I want to be honest, there's 2 pathways being tested here. Anything that strongly affected tetrapyrrole metabolism might influence the degree of lesions, right? If you could make less phototoxic metabolite, you would have fewer lesions. If you made more phototoxic metabolite, you'd have more lesions. So there's really 2 things being tested there. One, tetrapyrrole metabolism, and the other one is mutator-regulated gene expression. Anything that clicks the mutator transposon on, or any host function that turns that mutator transposon off, is going to modify the amount of lesions. And this is just to say, some of you may have seen this. I occasionally award certificates, the title of real maize geneticist for performing more maize crosses than Ivan Baxter and understanding epigenetic regulation of transposon activity. This was the one I think I gave to Abby in September. But if anybody wants one of these, you just got to go out and do a few crosses. Not many. Ivan's a lab guy. All right. So the variation in severity is awesome. So you can see here NC304 is covered in spots, and here NC 250 is not. And all of these contain the less 28 gene, less 28-1. And then here we have it, another replicate. This is just 2 replicates from the same plot of the same genotypes. You can see it's highly reproducible. So you could sit here and you could say this is 1 level of lesions, this is 2, this is 3, this is 4, this is 5, and that's a white box. Okay, so we can use this as a phenotype and do genome-wide association, and we did. And when we did, we got 1 spot of the genome that exceeded our genome-wide significance threshold. This is very likely to be true, right? P-value 10 to the minus 10. 1 chance in 10 billion that it happened by random chance. Little wrinkle, it's in the middle of nowhere. There's a huge section with no annotated genes, which initially seemed like a bummer. But we asked the question, are there any mutator-like sequences that might be regulating mu activity at this locus? And awesomely, the answer is yes. There is an unannotated, very well expressed locus here that encodes a mutator transposase, or a relative of the mutator transposase. Since the mutator family is quite large and it includes a group of elements called the vandals from Arabidopsis, and vandal, of course, is a pejorative term for an indigenous European group of people called Lugii. We decided to name ours Lugii. So Lugii turns out to be fascinating. So maize encodes multiple alleles of Lugii and multiple nonsense stop alleles. So this gene has been knocked out repeatedly within the lineage leading to Ziamase maize. About 17 inbreds out of the set we have whole genome sequence for, so 37% have a fully functional copy of this transposase, and the remainder have some deletion derivative, all of which should trigger nonsense-mediated decay and be loss of function. It's also expressed at very high levels in the lines that have the full allele, and expressed but at much lower levels in these consistent with nonsense-mediated decay kicking in and these being loss of function. In addition, there's multiple functional domains that are required for the activity of the protein that are encoded in the region past the stop codon. So, all right. Then it turns out, even better, we missed this gene in the annotation of the maize genome. This gene has been sitting in this location since before the divergence of Trypsicum dactyloides from the lineage leading to maize. So Lugii is a conserved synthetic gene, at least 650,000 years old, and possibly older. We're actually waiting for the Andropogon project to release their information. They got burned on a reduction in budget and COVID, and they haven't been able to get their paper out. So we're waiting for the next taxa here, but Toby Kellogg and Ed Buckler have promised they'll make that available. So Lugii splits, it's a real gene. It regulates mutator transposon activity, at least as reported by LES28 expression. And so we appear to have a natural variant that is involved in the epigenetic regulation of transposon activity, which I am super excited about. To take it a step further, we've been working with Jeremy Lohman at Michigan State University to try and do some biophysical modeling and try and figure out how this works. And so Jeremy has been playing around with, we know that these mutator transposon proteins, these transposases work as dimers. And we know that there are some sections in the terminally inverted repeat that these bind. And so Jeremy has been doing some biophysical modeling of DNA and protein interactions, and he got a really interesting and surprising result that matches the gel shift experiments that have been done previously on where these transposases bind to mutator DNA. And that is that it binds at two regions as a dimer, two regions that are separated by a gap, exactly as found in the gel shift assays, but it does so as a non end-to-end mirrored dimer. It's an unusual dimer form. These are fairly uncommon in nature. And when you think about like an endonuclease, which is what this is, typically you have two active sites that are oriented towards the phosphate they're going to cleave on each strand. And so they're in a symmetrical orientation. That is not what this is. It's also not the spot where it cleaves, but it might be the spot where it regulates gene expression. So I'm super excited about this. The biophysical modeling totally fits everything we've collectively generated in humans, but comes to a little bit of a surprise, and I think it's a nice hypothesis. So our model for how Lugii works is it's a dominant negative that binds to and sequesters the murA transposase, thereby altering mutator activity. In the case of the alleles that result in nonsense-mediated decay, it does not accumulate, and so the mutator transposase can function. And so we're looking at at a dominant negative inhibitor of mutator activity. And so that's what I think is going on. So less 28 is a Euro-S enzyme. 28-1 and 2 are suppressible alleles. High activity of the mutator results in lesions, and loss of that activity suppresses lesion formation, and natural variation uncovered a new Murray-like gene, which we think encodes a dominant negative transposase, but definitely encodes a transposase that split off and became a gene 650,000 years ago, that appears to suppress mutator activity. And now I'm at it. So with that, how about, whoops, that was not entirely right. How about some questions?
Well, I have a whole board, so.
Yeah.
Anybody have any questions?
So in the first part of your talk, you mentioned about using the unit of study should be either genome or genes, but not SNPs.
Well, for that particular approach, yes.
I was wondering if you could elaborate that a little bit.
So when you're doing this multi-species comparison, you're not asking, it's not the same SNP in soybeans and Arabidopsis. And it's not even just the same gene, but it's two sets of genomic regions that have been identified. You do a SNP that tells you this locus is important, and now I have to ask the question, what's my confidence interval? So it's going to be some region around that association. So that's what you're feeding the input to this combinatorial game are regions of genomes. So if you want to figure out what's the likelihood that I'm lying to myself, you also need to feed regions of genomes of a similar size into your permutation. If instead you feed into your permutation randomly selected genes, or you narrow your interval too much, or you try and regularize it too much to not match the sort of squirrely nature of genome-wide association experiments and the differences in LD between species, you go wrong, and you get these fabulous p-values because the mix and match did better than your bad characterization of your own experiment, right? So you're detecting a great separation because your model wasn't a good fit to your experiment. Does that make sense?
Yes, it does.
Okay, sure. Sharon, you had your hand up?
Yeah, so for the last part,
Yeah.
is the mutator element in the promoter of the lesion?
So there's with the two alleles we have, one, actually the mutator element is in the first intron. That's the strong one. And the other one, the mutator element, is in the promoter, the weak one. You're thinking about mechanism of how does this weird allele do that thing?
How's it do it?
Yeah,
We don't know.
we actually don't know. But since you've given me the opportunity to speculate, I think it's reasonable to think that these are fairly abundant genes. They're well expressed, they're tightly regulated. And there aren't that many genes that are mu suppressible, and there are a lot of genes in porphyrin biosynthesis that are mu suppressible. So I think there's something about the regulation of this pathway that is mu sensitive, but sensitive to the insertion of a mu element into its cis regulatory environment. So one possibility is that there is an RNA, and what we're actually looking at is aberrant RNA being created by the mutant allele having an effect on the wild type locus on the other side. Because we know from our monosomics that you can drop the expression by 80%, then you do not get lesions. So it has to be going further than that. The other possibility is that it's encoding a bad protein, that inserting in the intron means you only get a piece of the protein, but then that doesn't work for the one that's in the promoter. This is weird splicing, and like you can come up, everything has to be more complicated to fit. But an aberrant RNA feels like the fewest number of moving parts from where we're standing now. Like that's the hypothesis to be in my mind. Yep, yes.
Yeah, two, so one following up on that.
Yeah,
In the first intron, and you have a smaller protein, could that be alternative targeting, like something early in fulcrum biosynthesis going to different compartments, perhaps?
it could. I will say that, Bonnie Bartel did some awesome experiments on these, many years ago. And she actually showed that making transgenes that were improperly targeted just resulted in the not complementing phenotype, but did not have some other effect. Now, I don't know, how carefully you should look. She was interested in restoring metabolism, but so there is some work to suggest that that wouldn't be enough. Another thing worth saying is that there are multiple uncharacterized billion-year-old paralogs of these genes that appear to be involved. They respond to regulation the way other pieces of the pathway respond and have not been characterized. And I suspect that when people actually do the structures of these proteins in plants, that they're going to find out that they can operate as heterodimers and whatnot. And there may be some regulatory subunits, and that may be what's happening. Yeah.
You got a slide on there.
Yes.
You had something on there about 0 days since
Yes.
your last experiment, and you had data that you're not gonna show
Unless asked.
So I'm gonna bite.
So you're gonna ask. There it is. Salicylic acid accumulation is controlled by environmentally sensitive QTLs in Sweden and Italy. And we did this yesterday. And you can see we've got constitutive massive QTLs on 3 and 4, and environmentally sensitive QTLs, bottom of 3, only present in cold, bottom of 1, only present in warm. Huh? Good stuff. So yeah, there it is. I promise we'll eventually write a paper. Go ahead.
Still back to that new insertion.
Yes.
Does the insertion affect splicing or just affect expression level of the gene?
I don't know either answer.
So that should be easily addressed, right?
That's those molecular hypotheses. That would be the next thing to ask is how does that work? Unfortunately, some experiments that were done to try and get at that made some assumptions about the allele itself that turned out to be false. So we thought we had data that would be able to address that, and we don't, because we were incorrect about the genetic background of the original allele. But yeah, no, it's something we're interested in answering. Yeah.
Any other questions? Going up.
I have one question.
Yeah, do it.
You use multiple species for GWAS.
Mhm.
When we do GWAS, we use a specific reference, right?
Yeah.
Reference genome. When you have multiple
Sure.
species, how do you do this calling? Then you can compare it and do generalization.
Yeah, so I mean, I think so there's two pieces there. So one is how do we don't have to, how do I say this? This is not a comparison of SNPs, right? This is an output of regions in the genome that affect a trait in each of these species. And so the unit of comparison across species is, you can think about it as orthology of genes, and the SNPs are not important. Within each species, the SNP calls that got used to describe the genetic diversity of that species are what's being used for the genome-wide association output. So there's a within species piece of the workflow that is, SNPs, traits, associations, and mapping those associations to genomic regions and pulling the gene IDs from that. And it's those gene IDs and regions that then get added to the comparison, which gets back to sort of your question. And that is what is then getting compared across. So the unit of comparison, once we do the multi-species part, is the genes and regions. not the SNPs.
Okay,
Right.
Yeah, you work based on genes, right?
Yep.
And then
Yep.
Thanks.
Yeah, sure. It does open up a thorny problem about what does orthology mean, or how did we choose to define it in this case? We made some pragmatic choices that you can pick with. Yeah, we did.
Yeah, I was gonna ask you that. So sometimes it's just two bushes, right?
Yeah.
You can't establish one-to-one orthology. So you
And we
have
don't.
5 here and 5 here, and you just count them all.
Yes, so the way we sort of made the sausage is we decided that we would operate with orthogroups. And those orthogroups are gonna change depending on what species you include. So if you were to, I said it will perform better as you move further and further away. That's true in one sense. In another sense, the further out you move, the more genes are in each orthogroup, and the harder it is to get signals. So there's some interesting additional knob tweedling available as we continue doing this and as people try it with other traits. Yeah, Dan.
So the orthogroups are defined merely by reciprocal best blast hits and not synteny. Is that true?
You have to.
The second part is true. The first part is not necessarily just reciprocal blast hits. If that makes you feel better. It doesn't have to be a reciprocal best blast hits isn't the only way of doing it, but I think conceptually, yes.
So when you have hits that are observed across 5 species, are those synthetic across all 5? And when do those tend to be synthetic with the subsets of species that share the hits in this analysis?
So actually, if all 3 of the boxes were synthetic, we'd have equal levels of evidence for each one of them, right? So there'd be no reason to color one of them green. The fact is that transposition and aneuploid speciation happens enough in plants that we break up the synthetic relationships at these kinds of timescales, right? So there's certainly a lot of synteny between maize and sorghum. And so we get less ability to distinguish which of the genes at region X is the most likely causative if we just do this comparison, because they're all basically the same. But when we throw in Arabidopsis, that relationship gets split, and we end up with just one.
But when you do Manhattan plot, right? You
Yes.
use haplotype or
SNPs.
you use the SNP, right?
Yep.
But then how you compare different species together?
So the SNP then gets mapped to a location. That location has genes, and the linkage of those genes to that SNP allows us to say these genes are linked to that
But
SNP.
you do a SNP calling for different species, right?
Yes,
And
absolutely.
with different reference, right?
Yes, because they're different species. Yep. So the worldwide diversity of maize, the worldwide diversity of sorghum, the worldwide diversity of oryza, the worldwide diversity of Arabidopsis, worldwide diversity of glycine are all used for glycine, or Arabidopsis, or maize, to detect regions. Then those regions are mapped to genes, and the relationship, the evolutionary relationship of those genes to each other is what's modeled here.
Yeah, okay. Yeah. I confused
Got it.
very well.
Got it. Great.
All right, we
Cool.
probably thank Brian.